Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show. Today, bringing another really awesome guest involved in science and technology that's going to create a better tomorrow for many people out there. Today, we have the honor of being joined on the show by Dr. Thomas Chen, who is the founder, chief executive officer, and chief scientific officer of a company called NeOnc Technologies. They are a developer of these proprietary patent-pending platform that can ultimately transport pharma-based therapeutics directly to the brain without the normal restrictions of the blood-brain barrier, ultimately providing patients with more effective treatments. And they're developing a portfolio of therapies for brain cancer as well as other central nervous system disorders. Dr. Chen is a board-certified neurosurgeon. He's also the Director of Surgical Neuro-Oncology at USC, where he's also a tenured professor of neurosurgery and pathology. He graduated summa cum laude from University of Illinois, Urbana-Champaign, went on to do his medical degree, University of California, San Francisco, and then following neurosurgery training at USC, also did his Ph.D. In pathobiology, where his thesis was focused on the role of immunotherapy in malignant brain tumors. Dr. Chen is also a fellowship-trained spine surgeon. He's one of the few surgical neuro-oncologists in the country that specializes in spine cancer surgery, and he maintains a very busy clinical practice, both in surgical neuro-oncology as well as spine surgery, and has a research laboratory focused on glioma biology. Dr. Chen has published extensively on this area of biology and neurosurgery. He's on the editorial board for the Spine Journal, as well as on the review board for neurosurgery and the Journal of Neurosurgery, and he also serves on numerous national neurosurgery committees. A lot of very important topics to get into today. We are honored to have him with us. Dr. Thomas Chen, thank you so much for taking time to come on the show.
Thank you so much. Thank you so much for having me here.
Yeah, I'm glad you're here. I'm looking forward to the conversation. Tom, I would love to start off back in 1996. It is at this time that, as mentioned, you published your dissertation. The title was Modulation of Cytokine-Induced Adhesion Molecule Expression in Malignant Gliomas, where you go into the extremely complex pathobiology of way these really nasty tumors modulate, evade the immune response. You talk about the microenvironment, the microvasculature, and a lot of the targets that you point out in your dissertation 30 years ago are still extremely ripe when we look at brain cancer today, especially if one can get drugs through the blood-brain barrier. Take us back, if you would, to 1996 and a little bit of the work that started this all off.
Sure. I was basically interested in tumor biology, especially with gliomas at that time. And immunotherapy was really not on the horizon for really any tumors in any specific format. And so when we were looking at the immunotherapy for gliomas, we recognized that gliomas are basically what we call a cold microenvironment. And so this microenvironment is just not very suitable for a lot of the T cells and inflammatory cells that we think about today for harnessing for immunotherapy. And therefore, we were looking at what sort of modulators that we can do to make these tumors more immunoactive or hot, basically. And so what we did was that we focused on what's called TNF receptors, and TNF stands for tumor necrosis factor. And TNF has always been thought of as been pro-inflammatory, but it's really sort of a very molecule that's very concentration based. And so you can have TNF at very high concentrations, and it can be used to kill tumor cells. But you can also have TNF at very low concentrations, and it can be pro-inflammatory. And when it becomes pro-inflammatory, it can also have a pro-growth characteristic to it. And so what we were interested in is in knowing that, in terms of this one particular cytokine, TNF, because everybody has been focused on another cytokine called TGF-beta 2, transforming growth factor 2. And TNF, we thought, was very important from the standpoint of how prominent it displays when we look at tissue sections of gliomas. So when I take like the tissue that I removed from surgery, and we do immunostaining for it, and we look at how much TNF is within that microenvironment, we find that there's a lot of TNF there. Okay. And interestingly enough, at that time, we were also, the concept of having TNF receptors was getting worked into as to how does TNF bind to a cell. And so there was at that time, a TNF receptor 1 and TNF receptor 2. And what we found was that TNF receptors were not only present on the cell membrane, but they were also getting shed. And when they were getting shed, the question was that, are they getting shed? Are they doing a protective effect on the tumor cell? Like, for instance, if you had TNF and the TNF receptors were getting shed and it's binding to the TNF, is that used to sort of like immobilize or negate the effect of TNF on the tumor cell? Or when you shed it, does that have a pro effect in terms of making that TNF now more effective because now it's binded to something and therefore less of a possibility of having a destructive effect on the tumor. And that was the basis of my thesis. And what I found was that essentially, yes, glioma cells do secrete TNF, and they secrete both TNF receptor 1 and TNF receptor 2. And what we found was that TNF receptor 1 was the more dominant of the player compared to TNF receptor 2. And then 3, in terms of the binding, what we found was that it really has effects on both the from the standpoint of protection of the glioma cell from concentrations of TNF, and also it also has an effect in terms of making the TNF stay in the microenvironment longer. And what we find was that with the TNF, and whether it was protective or stimulatory, was essentially how much TNF was present. So you could add lots of TNF, like say, to a cell in vitro, and you'll find that essentially that there is a lot of TNF receptors shed, and there's less of an effect on the tumor cell. So there's a protective effect. But if you add a small amount of TNF in vitro, what you find is that you do have TNF receptors shed, but it's not nearly as much. And then the TNF receptor and the TNF effect seems to stay longer than just TNF itself. And so I think from that standpoint, the thesis was really important because of the fact that we're realizing that in all field of immunology now is that the immune system is really a dual-pronged system. It cannot really be just say, oh, I've got my immune cells there, or I'm doing immunotherapy, and therefore I should be, knocking the tumor cell out.
Mhm.
It really is not that simple. The tumor cells are very smart, and they will essentially season their own microenvironment to make it safer and more effective for them to progress further.
Mhm.
And of all the things that I sort of projected in my thesis, it was the fact that this is a dual system that we're working with, and we need to be cognizant of not only that there are multiple different cytokines present in our microenvironment, but they all have different roles, and what role exactly they play is very concentration dependent and very dependent on what's going on within the microsystem itself.
Excellent. Excellent. And so you developing your neurosurgical practice, you obviously have this incredible sort of encyclopedic understanding of the pathobiology of these tumors. They are, extremely multifaceted. And then along the way, and again, I spent some time sort of walking through your publications. You did not shy away over the last 30 years in trying everything, in the sense, you have papers on combination therapies, on repurposing drugs like protease inhibitors and COX-2 inhibitors, novel natural products, gene therapies, even, you studied things like fasting. We're going to get to the blood-brain barrier in a minute, but talk a little bit about your experiences here. Clearly, a lot of targets in these tumors that have, multiple personality. What are some of the top line learnings that came in the following couple decades with regard to mechanistic approaches to dealing with these tumors? And then we'll get to the blood-brain barrier in a minute.
Yeah, great question. And I'll tell you this, that if I were smarter, I and found out an exact way of treating these cancers, I would not have been doing all these multiple modality treatments. But the truth is that these gliomas are so different in terms of their mechanisms of resistance and how they can be targeted and affected. That I've always believed that there is no one silver bullet that's going to cure this cancer, and that's why we have always been doing out of the box work, trying to see if there's other modalities that may be involved. And I think that, as you mentioned, that I have been involved with gene therapy trials. I've been involved in development of gene therapy for gliomas. I've been involved in looking at COX-2 inhibitors for gliomas. And again, all these different ways of doing it target one specific pathway. And the question is that in terms of this one specific pathway, what kind of effect are we getting? And then is there a way of combining that with another pathway that may get us at least an additive or even synergistic effect? And that's really been the basis of a lot of the work that I've been doing. And so I would say this, that in terms of my 30 some years of treating gliomas, the only thing I can say is that I've been very humbled by it.
Understood. But no, you're making a lot of progress, and one other place I want to go before we get to what's going on in NeoOnc is the blood-brain barrier, because here we have the nastiest of nasty tumors in terms of the gliomas and the glioblastomas and so forth, and we're behind this semi-permeable barrier, about a quarter of a millimeter thick, and it does a great job of keeping out small molecules and proteins and microbes and everything else. Talk a little bit about the blood-brain barrier and what are the properties that make this, from a non-scientist look at it, doesn't seem it should be so hard to get stuff across this thing. walk us through a little bit of that, and then we'll get into what NeOnc's up to.
Sure. Okay. Well, first of all, the blood-brain barrier is made up of cells from the brain. And so we have something called the blood-brain barrier because of the fact that it is very, very crucial for us to have a blood-brain barrier to prevent small molecules, viruses that we do not want to get into the brain. And so it's a very important protective effect. If you think about the cells of the brain are really neurons, astrocytes, and endothelial cells. And the blood-brain barrier is mostly formed from astrocytes and endothelial cells. And the endothelial cells are the cells of the blood vessel. And what this barrier does is that it allows certain molecules to get across, and for the most part, it prevents other molecules from getting across. We do know some important characteristics about it, and one of them is that if you are more lipophilic or fat soluble, you have a better chance of getting across the blood-brain barrier than something that is hydrophilic or water soluble. Now, most of our compounds that we have are hydrophilic. They are basically water soluble drugs, and so that's why you're able to get drugs intravenously. That's why you take drugs orally, and then they get distributed throughout the body. And the thing is that with the blood-brain barrier is that it is preventing drugs that we take intravenously or orally from adequately crossing it to get to the target. And when we have a brain cancer, the target is in the brain itself. And so question is that how do we allow that to get across? Now, this is a very, very important topic for pharmaceuticals in general. And many pharmaceutical companies are trying to make their drugs more blood-brain barrier soluble. And so one of the ways they've been doing is essentially they've been designing their drugs so that they are more lipophilic or more fat soluble, so that it's easier to cross the blood-brain barrier. Now, throughout all my studies that I have talked about, one of my biggest frustrations was essentially how do we get drugs across the blood-brain barrier better? And so we came across this concept of nasal brain delivery. And nasal brain delivery, what we're doing is that we're not trying to cross the blood-brain barrier. We're not trying to, make a compound more lipophilic. Well, all we're doing is we're saying that let us use our existing cranial nerves, and allow the cranial nerves to deliver the drug to the brain. And so we're just essentially bypassing the blood-brain barrier. And this technology has actually been very well worked out in animal models. And as a human being, you know that this delivery exists, because what has been found is that the first and the fifth cranial nerves are the two main conduits for delivery to the brain. Now, we have 12 cranial nerves in our brain. And some of them have pure motor function. Some of them have both sensory and motor function, but they all have a role. Now the first and fifth cranial nerve is the olfactory nerve, the smell nerve, and then the trigeminal nerve, the nerve that provides sensation and allows us to chew. Now, let me give you the example of the olfactory nerve. You, as a person, know that if you smell something, that scent goes to our brain. Our brain recognizes it. We know it's there. So somehow that scent molecule is able to go to our olfactory nerve, goes to our olfactory bulb, goes via the olfactory tract, to the mesial temporal lobe of our brain, and we recognize what that scent is. Okay, so you know that smell is almost instantaneous, and you know how rapid that can occur. And imagine this: if we had to go through our blood-brain barrier to allow that molecule to cross, and then for us to decide what it smells like, it would take probably a little while for us to recognize what we're smelling, right? And so what we're doing with the nasal brain delivery is we're asking a question. Okay, so this nerve allows molecules to come to it. It basically senses what's there and what's being delivered. So the question is that, can we accessorize that nerve so that we can essentially inhale our drug to the brain? Okay. And that's what our nasal brain delivery technology is doing. Now, we are not the first company to be talking about nasal brain delivery. We are the first company to use this technology for treating brain cancer. Right. And so one of the things, so one of the things that people have always asked me is that, okay, so if I have a cancer, and the cancer, let's say, is in the parietal lobe of the brain, so it's up on top, and so you told me that the olfactory nerve essentially allows you to conduct smell to the mesial temporal lobe. So what is it that when you get the drug to the brain, how is that drug able to go to the target, which may be separate from where it normally would be going for smell? And so we try to answer that question both in animal experiments and in humans. And so in animal experiments, what we have done is that we have taken our drug, it's called NeO-100, and we have given it intranasally to mice. And then we have done biodistribution studies of this drug for the brain. And we find that essentially that the biodistribution of the drug to the rest of the brain is excellent. It essentially will allow this drug will be able to permeate through the whole brain. And we believe that the reason why it's able to do that is because of something called the lymphatic system or CSF conduction. Yep. So when the drug gets passed through the cribriform plate, we believe that a lot of this drug basically is carried by the cerebrospinal fluid through the interstitial spaces of the brain parenchyma to the target. And what we have done is that we actually have verified that in a patient. So one of my patients was on phase 1 trial with NeO-100. And she was doing this intranasal treatment 4 times a day. And what happened to her was that her tumor recurred, but she had been getting treatment for over 3.5 years now. Okay. So when her tumor recurred, I told her we should go back and resect it. Okay. And what we did was that we had her inhale NeO-100 for 1 week prior to surgery. And then at the time of surgery, I removed the recurrent glioma. And then we submitted that to tissue analysis. And we did HPLC analysis to see how much NeO-100 was present and how much its active metabolite, perillic acid, was present in the tumor. And then, if that was present in the tumor, this would lead us to have verification that the drug was getting to the target. And we were able to demonstrate that, and we published that. And now I also want to add that there's probably no more direct way of verifying that it gets to the target via molecular analysis. But our other way of analysis, even though it's broader and more crude, is just MRI scans.
Sure.
MRI scans showing that, oh, the patient is not getting treatment by any other means, but via this intranasal delivery, and the tumor is getting smaller. Well, tumors don't just get smaller by themselves.
Right.
So this is also another way of verifying that yes, the drug does get to the target. Mm-hmm. Now I've got big dreams about this drug, because we believe that this drug at high concentrations, and I guess this gets back to my TNF work, at high concentrations, it has activity in terms of getting the tumor smaller and also decreasing its proliferation. And NeO-100 does slow tumor growth down. It's a good cell cycle inhibitor. It essentially blocks the tumor in one of the cell cycle growth phases, but also induces a mechanism called ER stress, where essentially it puts pressure on the tumor cells and then can lead to apoptosis or cell death. Mm-hmm. Now, with NeO-100, what we also found is that it's actually a very good vehicle for transport. So we did some experiments where essentially we took molecules that would not normally pass through the blood-brain barrier, and we put it in NeO-100, and we demonstrated that that drug was now able to get to the brain. Mm-hmm. And let me give you an example. Velcade is a proteasome inhibitor that is used as a second-line therapy for multiple myeloma. Now, if you use Velcade and place it into in vitro in a dish of glioma cells, you find that it is very, very active. In fact, it can kill glioma cells at what we call nanomolar concentrations. Now, usually, if we see micromolar, it's pretty good, but nanomolar is excellent. Yep. And because of this activity that was reported in vitro, some of our neuro-oncologists have done clinical trials given Velcade intravenously to patients with glioblastoma. And what they found was that it had absolutely no effect. And again, the question is why? So what we did was that we explained why, because what we did was that we implanted a tumor into a mouse brain. We gave Velcade intravenously, and we found that, as in the human studies, there was no effect. And our hypothesis was that, well, maybe the drug is just not getting to the target because it's not getting across the blood-brain barrier. And so what would happen if we took Velcade and put it in our NeO-100 and gave it intranasally? Would that have an effect on the tumor that's growing in the mouse brain, in the brain? And what we find was, yes, it was actually that the tumors were getting smaller and the mice were living much longer because of the fact that the Velcade was now delivered intranasally than intravenously. Mm-hmm. And imagine what this does for you in terms of drug delivery to the brain, is that now you're not trying to I'm trying to make Velcade more lipophilic so it can cross the blood-brain barrier better. No, what we're saying is that if you have a drug that works, whether it be immunotherapy, whether it be targeted therapy, whether it be small molecule, whether it be microRNA, we can test it by dissolving it within NeO-100 and delivering it to the brain. Now, one question you may have is that, well, why don't we just put it and just sniff it up? And why would NeO-100 even have to be involved? Okay, and so what we have done is that we have done experiments where we essentially take our molecule of choice, like let's say Velcade, or drug of choice like Velcade, and we gave it intranasally without NeO-100. And then we compare that to giving it with NeO-100 for what we believe is increasing the efficacy of delivery across the nasal brain delivery pathway. And we find that it is much more effective when that drug is combined with NeO-100 to go through this intranasal brain delivery. Mm-hmm. And we actually have done this in mouse experiments where essentially we have put in antibodies, we have put in small molecule drugs like microRNA, and we find that it's actually able to deliver this drug or therapeutic to the brain in much better ways than the therapeutic itself would be able to get across either intravenously, orally, or just by itself intranasally. Mm. And that's where I think the value that NeO-100 is going to be, that we understand that basically at low concentrations, it can be used as a solvent for delivering other drugs to the brain. At high concentrations, it by itself can be used to impede or kill tumor cells that are in active growth.
So it's it's quite interesting. Just I mentioned this to you when we were chatting before the show, but I spent a couple careers ago, I spent time in the natural products drug development space, and I know Perillyl alcohol. At the time, we were isolating it from cherries, strangely enough, for what we were doing. But again, so just for the audience, so you have a wonderful delivery vehicle. You have a wonderful delivery vehicle that also has its own quite interesting anti-cancer effects. And then what you're doing, you're combining it with a range of substances, everything from temozolomide, sort of the gold standard in glioblastoma therapy nowadays, to a range of large molecules. So yeah, I mean, it's a beautiful package. And again, the natural products just add another interesting little part of the story, which I'm very passionate about. Talk a little bit about, because you mentioned, okay, so the core substance is NeO-100. There's a series of other conjugations like NeO-212, which is the temozolomide combination, and then a series of clinical trials planned: glioblastoma, malignant glioma, you have meningioma work. Walk us through a little bit of, I mean, obviously a lot going on, and these are all unmet medical needs. Walk us through a little bit of what's happening on the clinical front today, if you would.
Sure. So, so right now the company has 4 trials that are in different phases of development. Our lead asset is called NeO-100-01, and that clinical trial is now in what we call phase 2A. Mm-hmm. And so that means that we have gone through a toxicity study with phase 1, and now we're in a phase 2A phase where we're trying to demonstrate efficacy, and in a small number of patients. Now what we have done in phase 1 is that we enrolled a total of 12 patients. These were what we call recurrent glioblastoma. So these patients have gone through what we consider is standard care treatment for a glioblastoma, and that is surgery, radiation, and temozolomide or TMZ. And temozolomide is our standard care drug. It's an oral pill that's given, mm-hmm. and it basically induces DNA methylation on the cancer cell. Now, these patients, once they have failed the standard care treatment, they have several choices. One is that they can use another drug that we consider second-line therapy, and this other drug may be a drug called Avastin, which is trying to be used as an anti-androgenic drug. Mm-hmm. They can go on a clinical trial. And some of these patients actually undergo another surgery. Now, when they go on clinical trial, they basically have to, essentially use the clinical trial drug as their sole source of treatment. And so when they came on our clinical trial in phase 1, these were patients that have failed the standard of care. The time of survival for these patients is about 6 to 8 months. And that's to be, just to be frank with you. And to be honest with you, in my practice, I never tell patients, oh, you have only 6 to 8 months to go. Okay. One, I'm not God. I can't predict how long somebody's going to live for.
Sure.
And then two, no matter how dire the circumstances, patients do better than that 6 to 8 months statistics. And so for me, the concept of hope is very, very important for these patients. And so when they came on our trial, I told them that this is a trial that we're initiating as a phase 1. You're going to be enter into a dosage that has been predetermined, and we are going to be dose escalating. Okay. Now, what we found was in this trial that, one, this is a toxicity trial, so none of these patients had any significant side effects. So meaning that, oh, did their bone marrow get affected? How was their liver? How was their kidney? How did they feel? The patients were doing great. They had none of these systemic side effects that you would anticipate with chemotherapy. Now, the other thing we found was that in these patients, we had 4 patients that actually lived for longer than 3 years. Okay. And so the thing is that, oh, you say, well, you only have 12 patients, and 4 of them did really well. And my response is yes, and I wasn't expecting them to be doing so well. I was, the trial when we started, it was only written for a 6-month follow-up for outcome reader on it. And so when these patients did so well, we asked the question, what was the common characteristic of these patients that they would do so well with this type of therapy? And so we did a genomic analysis on these 4 patients' cancer, and we found that they all had a mutation called IDH1 mutation. And when we found that IDH1 mutation in these patients, and we were at that time we're starting to enter phase 2A, we wrote to the FDA and said that, we have patients that have done much better than we anticipate. Do you think when we do our phase 2A that we could enroll just these patients with the IDH1 mutations? And the FDA gave us a go-ahead to do that. Now, the phase 2A is enrolling a total of 25 patients. And we are actually done with enrolling for the majority of the cases. We basically have about 7 more patients to go, and then we'll be closing the trial for phase 2A. And that's where we are with our lead asset, NeO-100-01. Now, we also have a clinical trial using NeO-100, and this is a phase 2 trial. It's for patients with unresectable meningiomas. Okay. Now, meningiomas are the most common benign brain tumor, but on the other hand, they can also become malignant, or they can also become unresectable because of their location. So a lot of these meningiomas are in what we call the skull base. So they're at the base of the brain. And at the base of the brain, we have many important cranial nerves. We have arteries running through there, making it almost impossible to get a complete resection. And so these patients with skull-based meningiomas are currently getting treated, for the most part, with just radiation. And so NeO-100-02 is for malignant or unresectable meningiomas. And our hope is basically to be able to make their meningioma a chronic disease. So instead of having these skull-based tumors slowly grow, and then patients have to go back to surgery, and some of these patients actually have gone back to surgery 3 or 4 times. And the question is that if they were to get this as a treatment, could we prevent that growth from occurring so that they do not have to keep on having repeat surgeries? And so that's NEO-100-02. Now, NEO-212 is our other therapy. NEO-212 is taking temozolomide, and in this case, what we do is we conjugate it, or essentially do a unique new chemical bond. In this case, is a carbamide bond of the temozolomide to NEO-100, and ask the question is, what happens when you chemically conjugate that? What happens to the properties of temozolomide? Now temozolomide, or TMZ, is something that's called prodrug. So this drug essentially, for the most part, breaks down in the bloodstream. And the active ingredient of this drug is a molecule called AIC. The AIC is the compound that actually goes to the cancer cell nucleus, and induces the DNA methylation that we consider as the mechanism for how TMZ works. But imagine this: if you had temozolomide and you're taking this drug orally, and I just told you that it's a prodrug that gets broken down the bloodstream, a lot of this active drug actually never gets to the tumor. And so some of it does cross the blood-brain barrier, but a lot of it just gets distributed to the rest of the body. And that's one of the reasons why, when you take TMZ, you can have something called myelosuppression, where essentially it knocks out your bone marrow. Okay. So this is one of the rate-limiting factors for patients taking this drug. Now, what we found is that when you conjugate it, it converts the temozolomide from a prodrug into a stable drug. And when you convert it to a stable drug, what it does is that it allows that drug to then cross the blood-brain barrier, and then it goes to the cancer cell itself. And then in the cancer cell, in the cytoplasm of the cancer cell, it then gets broken down into TMZ and the para-alcohol, or the NEO-100. And that TMZ then gets broken down into AIC, and then it goes to the cell nucleus and then kills the tumor cells. So we have done proof of principle of this in animal models, but much more gratifying is our NEO-212 trial. Because what we've done is that with our NEO-212 trial, we've taken this compound, we've been giving it to the patients that have failed TMZ already, and we give this compound to these patients, and we find that it's very well tolerated, and it has effects on their brain cancer, even though they have failed TMZ treatment. And the other thing that is probably almost more important is that when you take TMZ, you can measure AIC concentrations in the bloodstream. When you take NEO-212 and you measure AIC levels, it's at least 1/100th the level of what you get with TMZ. And that's sort of substantiating what we're seeing in these patients. That essentially that they are not having any bone marrow suppression with taking our drug. And we believe the reason is that the drug does not break down in the bloodstream. We're able to stabilize it and enhance its delivery to the tumor. And also the other thing we did was that we did lipophilicity profiles of TMZ and our drug NEO-212, and we also find that by conjugation, it makes the drug more lipophilic or more fat soluble. And we believe that's another reason it crosses the blood-brain barrier better. And so NEO-212 now is undergoing clinical trials predominantly for patients that have a primary brain cancer and that have failed standard care. Now NEO-212 in our animal models also does very well in treating brain metastases.
Okay.
Now, if you are to ask me, like, well, how many, primary glioblastomas are there in the United States per year? It's about 18,000 cases. Okay. If you were to ask me how many patients with cancer have brain metastases, it's about 225,000 cases per year. Okay. So it's an order of magnitude in terms of primary versus metastatic brain cancer. Now, metastatic brain cancer right now, if you have a metastatic brain cancer, you undergo surgery and you undergo radiation. But because of the blood-brain barrier, many of the drugs that these patients are getting, whether it's for lung cancer, breast cancer, they actually don't do very well in terms of treatment of brain metastases. And so what we have been working on is essentially putting our NEO-212 into a carrier vehicle, such as a liposome or a nanoemulsion, and essentially allowing that NEO-212 now to be not only delivered orally, but to be delivered intranasally. And we hope that by doing this, we will be able to target brain metastases with NEO-212 and help these patients with brain metastases where essentially they don't really have a chemotherapeutic option for treating their metastatic brain cancer. And the last trial that we're doing is also related to NEO-100, and it's for children. I don't know if you know this, but children, like anything else, can get cancers in the brain. And unfortunately for our kids, the cancer in the brain is usually not in a very good location. And one location that is very deadly is called a pontine glioma. So they can get a glioma in the pons of the brain. And the pons is in our brainstem. And so when these kids get a pontine glioma, they currently are treated predominantly with radiation. And there are some chemotherapeutic drugs that are getting developed that may have an effect. But the bottom line is that these children have a very poor prognosis. Now, what I thought would be interesting is this, that I told you that the first and the fifth cranial nerves are the main conduits that we believe works with intranasal delivery. So I had a hypothesis, and our hypothesis was that, okay, so the fifth cranial nerve actually emanates from the pons itself. So could we take children with pontine gliomas and essentially allow them to do a non-invasive therapy like inhalational therapy and inhale their therapeutic to the pons and affect the pontine glioma? Okay. And so that's what we have been working on. We actually submitted this protocol to the FDA, and we received approval to run a clinical trial for these children with NEO-100. And we have then contacted, there's a large consortium called the Pediatric Neuro-Oncology Consortium, or PNOC. And we have contacted them about doing a trial based on our IND approval for these patients with pontine high-grade gliomas. And along the way, we actually have been doing some compassionate use treatment for some children with pontine gliomas already with NEO-100. And we're finding that these kids, they're usually about range from 6 to 10 years old. They have actually been inhaling NEO-100 and actually have been doing very well with their inhalational treatment in terms of toxicity. And we're finding that essentially not, hopefully, this is obviously too early to talk about efficacy, but these children have been doing well with this type of treatment. And we hope to essentially standardize that in a formal clinical trial with NeOnc, with inhalational therapy for kids with high-grade gliomas in the brainstem. And so these are our 4 clinical trials that we are currently undergoing, and we hope to essentially not only talk about developing a new way of treating brain cancers via this intranasal route, but also new molecules such as the NeO-100, which you've talked about as basically a purified version of para-alcohol that you had worked with before, and also a conjugation, the NeO-212, that can be used for treatment of brain cancers. But what I want to say is this, that we are in the process of developing not only the delivery method that may be effective, but the new drugs. But think of this, that if we prove efficacy in the worst of the worst, which is the brain cancer, imagine if what we can do exploiting this technology for neurodegenerative diseases, for instance. It would be amazing in terms of what can be done to help people with brain disorders. And so in a way, you know, people ask me, so why are you doing brain cancer when you could be doing something like, you know, Alzheimer's or Parkinson's? And my answer is that, well, I'm a neurosurgeon. This is my field. This is my expertise. This is how I want to help my patients. But I also understand that it's possible that if we are able to demonstrate efficacy in the worst of worst conditions, like a brain cancer, that we can apply this to myriad conditions of the brain itself.
Yeah, the CNS possibilities outside of oncology, whether you're talking Alzheimer's or Parkinson's or various other diseases. I mean, yeah, the potential is unlimited there, and it's a fascinating, you know, therapeutic story, but at the same time, it's an extremely impressive drug delivery story. Thomas, while we have you, I had to ask, and then again, this goes back to your dissertation work and sort of the immunobiology of tumors, but I couldn't help not see this paper that you published this was December 2023 in the Journal of Neurosurgery, entitled "Enhancing Brain Entry and Therapeutic Activity of CAR T Cells with Intraarterial NeO-100." This is in a mouse model of CNS lymphoma. Could you say just a couple words? I mean, I know that's not part of the clinical program right now, but obviously CAR T and immuno-oncology is such a hot space now, and some of what you saw there in terms of, I mean, that's one of the holy grails in CAR T cells is how we ultimately improve delivery of these things for the solid tumors. Can you say a couple words about, I mean, I don't know if it's confidential or I know it's early stage, but if you could talk about that.
Sure, it's early stage, but it's something that we're very excited about. And to be honest with you, from a clinical bandwidth, we just do not have the clinical bandwidth of doing this right now. But what we found was that the NeO-100, basically what it does is that if you give it intra-arterially, that it can actually physically open up the blood-brain barrier for about 4 hours, and then the blood-brain barrier reforms again. And so what we do is basically we are currently doing these experiments in a rabbit model. But what we can do is that, I don't know if you have heard of people with aneurysms in the brain. They currently undergo coiling for their aneurysms. So when they do these techniques, the interventional neuroradiologists, they currently undergo catheterization of either the femoral artery or the radial artery. And then they're able to snake that catheter all the way up to the neck where the carotid artery is.
Mm-hmm.
They're able to advance it to one of the arteries in the brain, whether it's the anterior cerebral artery, middle cerebral artery, and then they're able to embolize a target in the blood vessel, such as an aneurysm. Now, what we asked the question is that could we essentially do this technique that is already FDA approved and it's widely used, and it's basically now almost a standard of care for the treatment of some of these aneurysms. Could we use this same technology, but essentially what we would do is deliver NeO-100 and then open up the tumor um, brain barrier and then essentially be able to deliver molecules such as CAR T cells to the tumor.
Yeah.
And so what we did was with this animal model is that in mice, it's essentially too difficult to do the standard catheterization. So in mice, what we do is the intra-arterial delivery via the atrium of the heart. And what we're able to find is that if you give NeO-100, it opens up the blood-brain barrier. And not only does it open up the blood-brain barrier, it allows molecules of different sizes to cross it. And so we started off with a drug that doesn't cross the blood-brain barrier. It's called doxorubicin. And we demonstrate that we can get doxorubicin across the blood-brain barrier. Then we went to an antibody, such as Herceptin, and we demonstrate that we could get an antibody across the blood-brain barrier. And then after we did that, we also did antibody drug conjugates and demonstrate that we can get that across the blood-brain barrier. And then the last one we did was the CAR T cells, and we demonstrate that we could get the CAR T cell across the blood-brain barrier to the target. Now, I would say that in terms of the CAR T cell therapy, that people ran up through were doing clinical trials with recurrent glioblastomas, and they were trying to give CAR T cells intravenously to these patients. And what they find was that there was no benefit to giving CAR T cells. And that's because CAR T cells, are not able to cross the blood-brain barrier. Now, what people are now doing with CAR T cells with brain cancer is that they're delivering it intraventricularly. So they're delivering into the cerebral spinal fluid so that it gets into the brain. And then hopefully that the CAR T cells will then migrate to the brain cancer and then have an effect. And so the thing about the CAR T cells is twofold. One is that it has to get to the target. And we're hoping with opening up the blood-brain barrier directly, we can get something large like a CAR T cell into the microenvironment. And then two, once it's in the tumor, it basically is going to be meeting a cold microenvironment, meaning that it's going to be meeting an environment that it's going to be very immunosuppressive. Yep. And so many of these CAR T cells essentially run out of steam in terms of getting to treatment for the target. And so there's two things that need to do to make it for the CAR T cells to be effective. And I think that we are in the front, at least getting the CAR T cells to the target. Yeah. And then we'll need to have more science to make them more effective once they actually get to the target. Yeah. But brain cancer is like any other cancer. It's like a melanoma, which has very good effects with CAR T cells. And that is that it can work. It's not that CAR T cells cannot work. It's just that we have to perfect it so that it can work at its maximal capacity.
Mm-hmm. Mm-hmm. Yeah, no, it's again, it's just another example. I know it's outside the bandwidth now with everything else have going on, but it's just another great example of ultimately what you put together with NEO 100, and yeah, again, there's the sky's the limit here. So I'm quite excited for you and
Yeah,
the company. And clearly, everything you built up over the last 30 years is, it's all very synergistic and coming together. Tom, what else is going on that we should know about for 2025? In terms of, I know you have a public offering, I think, coming up at some point. Conferences, talks, other public facing stuff that's happening that you want to mention. Take the floor.
sure.
Anything I missed?
Sure. So let me address the public offering first.
Mhm.
The public offering is actually going to come very soon. It's going to be listed. We are going to be listed on Nasdaq Global, and we basically are doing this through something called direct offering.
Mhm.
And we hope that the public offering will essentially enable us to get funding to carry out and accomplish our clinical trials. We are, in terms of our readout, we are going to be more advanced than most biotech companies coming out. Most
Yep.
biotech companies don't have readouts until 3 or 4 years later, and they're working
Yep.
on, clinical trials or preclinical data. We're
Mhm.
going to have some readout in our phase 2A very, very soon because we only have 7 more patients to recruit to finish it. And then we're going to be closing our NEO-212 very soon because we're already on cohort 4. And
Mhm.
so we're going to be giving the public readouts in terms of our clinical trials very soon. So we're not going to be like a typical biotech company
Right.
waiting for data for a long, long time period. And then the other thing is that in terms of presentations, I regularly present at something called American Association of Cancer Research for our
Mhm.
research. We will be continuing to do that. We'll also continue to present at what we call our Society of Neuro-Oncology conferences. And then the following year, we're going to be submitting abstracts to ASCO, the American Society of Clinical Oncology, in terms of our clinical results that we have obtained. And so, from a scientific academic standpoint, we have a number of presentations that will be outgoing. And then, from the standpoint, one of my things that I want to do is that I want to increase our social media presence for the patients because you know that when clinical trials come out, different investigators have different interests in terms of what trials they want to be advocating. And patients, oftentimes are going towards whatever clinical trials their investigators propose. But
Mhm.
what I want to do is I want to just get the word out to the patient that there is a trial that, such as like NEO-212 and NEO-100, that can be used to give them hope and potentially extend their lifespan that is available and that we feel can be effective. And so I would like the patients to be more informed, and I would like the patients to ask their doctors, "Have you heard of NeOnc and what they're doing in terms of their treatment?" So that we can get more awareness of what we're doing, because I really think that we are on the cusp of having something very unique and very important to helping patients with brain cancer.
I concur. I'm excited for you. I hope I can help you spread that message and get the word out there with this episode. But it's really impressive work, Thomas, and just want to really again thank you for taking the time to walk us through all this, and really wishing you and the team the best in 2025 for the 2026 readouts. Again, for everybody that is going to be listening to this episode of the show across the various podcast networks or watching on the YouTube channel. You've been spending time with Dr. Thomas Chen, founder, chief executive officer, and chief scientific officer of NeoOnc, director of surgical neuro-oncology at USC, professor of neurosurgery and pathology as well, doing really amazing things to create more effective safer treatments for patients with brain cancer and other CNS disorders. Thomas, again, I want to thank you for taking the time out of your schedule for a while to talk to us, explain everything you're up to. Obviously, thank you for what you do for all these patients. And as we like to say on our show, again, thanks for creating a better tomorrow for all of them via the type of work you're doing and continue to do. Really awesome.
Thank you so much.
Great having you.